In this video, we will discuss about transcriptome analysis of the various ex-vivo models that are available to grow pancreatic cancer cells and compare them with the human pancreatic tissues results. In the previous video, we have validate the custom designed PA-ECM in vitro, whether they are able to culture the pancreatic cancer cells or not, and we have find out that they are able to culture in the PA. The researchers are able to culture pancreatic cancer cells in the PA-ECM matrices, and its behavior looks similar to the human pancreatic tissues in terms of the phenotype. And in this video or in this experiment, in this figure, they have compared the transcriptome analysis of the PA-ECM and the other ex vivo models and compare them with the human pancreatic tissue. In the panel A, we can see that the comparisons between three different pancreatic cancer affected patients and the other ex vivo models are shown here. In this case, we can find out that the in vivo results are in line close to each other, and similarly the other ex vivo results of the different approaches are lying close to each other in most of the cases. And one more difference that they have find out is that ex vivo results are slightly different from the in vivo results. It can be seen clearly in this figure. And they have also plot the PCA and they have also plot the PCA plot for this data that can be seen from the supplemental figures. And the reasons that they have hypothesis that could be because of the difference in the difference of the matrices that are present in the human pancreatic tissues and their ex vivo models. And after that, they check the gene analysis on the different samples. What they have find out is the gene analyses are almost similar to the in vivo and ex vivo models that can be seen in the panel B. And thereafter, what they want to see, they want to check uh, what are the difference, what are the reasons for the difference between the ex vivo models and the in vivo models. In order to do that, what they have hypothesized that these differences could be because of the matrices that are present in the human pancreatic tissues and which are mostly absent in the ex vivo models. In order to compare that hypothesis, what they did is they analyzed the two different ex vivo approaches. One is in the organoid form, and another one is in the PA-ECM that has matrices similar to the or close to the human pancreatic tissues compared to the other ex vivo models. And what they have find out is that the genes that are dysregulated when they compare between these organoid ex vivo models and the PA-ECM models, the genes that are dysregulated are mainly related to the matrices or that are affected by the matrices interactions or extracellular matrix interactions. After that, they also check the pathways that are dysregulated in the different ex vivo models, and what they find out is that in the PA-ECM, the dysregulations, the pathways, pancreatic cancer cells behave more similar to the basal subtype, and which is also in line with the patient's conditions that are used for the analysis, whereas the other models such as organoid behaves similar to the classical subtype. Subtype, uh, organoid or spares behave similar to the classical subtypes indicates that these ex vivo models uh, underrepresents the actual pancreatic cancer cells, underrepresent the actual pancreatic cancer patients conditions, and is only represents the uh, classical state of the classical subtype of subtype or general conditions of the pancreatic cancer patients, or we can say the initial conditions of the pancreatic cancer patients. After that, after validating that the PA-ECM models is more close to the in vivo conditions of the pancreatic human pancreatic tissue that are affected with the cancer, they further validate their results using the using the correlation coefficients, where they have find out is that the stem cell factors in the other ex vivo models are underrepresented compared to the case of this PA-ECM, where stem cells are represented more. And however, these core ECMs are least represented, and the reason because why this core ECM is least represented is because core ECM is mainly been affected by the matrices, and this, even though this PA-ECM has some matrices, but its matrix compositions is quite different from the actual human pancreatic cancer cell affected patients, and this is the reasons the core ECMs are represented least, both in the not only in the PA-ECM ex vivo model, but in the other ex vivo models also. And also this epithelial cytoskeletal, the expressions are better in the PA-ECM than in the organoid. So from this correlation coefficients also, they further substantiate their uh, custom designed PA-ECM, that it provides a better in vivo it provides this what they have finally concluded is that the PA-ECM provides a better in vivo model compared to the other is a better ex vivo model that can replicate the in vivo uh, characteristics compared to the other ex vivo models that are available in the market. So I hope you understood basic comparisons of the transcriptome analysis between various ex vivo models and the human pancreatic tissues, and how they have managed to say that PA-ECM is is a better ex vivo models compared to the other ex vivo models for pancreatic cancer cells representations. That's all for this video. And if you need to understand more about these results in detail, you can go through the reference that has been provided in the description. Thank you for your time.